Assalamualaikum to all of you. I am Dr. Hadee shagird. Let's start our topic without consuming our precious time in useless chat. In the previous lecture, we have discussed about the transcription, in which messenger RNA has synthesized. Now we will discuss about the translation, in which use of messenger RNA will occur. Okay. So what is the definition of the translation? The formation of protein by the ribosome is called translation. It is the simplest definition of the translation. The modified definition of the translation is that conversion of sequence of nucleotide into the sequence of amino acid by the ribosome is called translation. It is the modified definition of the translation. Now we are going to discuss about the ribosome, which are responsible for the translation. Okay. Ribosome are the tiny granular structure which are present in the cytoplasm. It may be in free state or attached state with the endoplasmic reticulum. They are also called proteins factory of the cell. Okay. Ribosome consists of two parts, two unequal parts. One is small and other is large. If we talked about the ribosome which is present in the prokaryote, it is 70S. If we talk about the ribosome which is present in the eukaryote, it will be 80S. Okay. Prokaryotes contain ribosome of 70S and eukaryotes contain ribosome of 80S. If ribosome of prokaryotes disassociated to two parts, large and small part, the large part will be 50S and small part will be 30S. If the similarly the ribosome of the eukaryote disassociated to two parts, large and small part, large part will be 60S and small part will be 40S. Okay. What is meant by 40S, 60S, 80S, 30S, 50S and 70S? We all know that during the centrifugation, different organelles of the cell begin to sit down to the bottom with a specific speed according to their weight. Similarly, during the centrifugation, ribosomes also sit according to their weight with specific speed. That speed is 70. Okay, the ribosome of 70 sit down with the speed of 70. That's why it is 70. If during the centrifugation, ribosome disassociated to two parts, the large part of the ribosome of the prokaryote will sit down with the speed of 50, and small part of the ribosome of the prokaryote will sit down with the speed of 30. That's why it is 30, it is 50 and 70. Okay. Similarly, this procedure occur in eukaryotic ribosomes. Okay. Eukaryotic ribosomes sit down with the speed of 80s. If it is disassociated during centrifugation, its large part will sit down with the speed of 60s and its small part will sit down with the speed of 40s. Okay, that's why these are 40s, 60s, 80s, 30s, 50s and 70s. Now I tell you about the function of the ribosome. Ribosomes are the factories of the protein synthesis factories. Okay. And they also form polysome. What is polysome? When we need rapid synthesis of the protein, then they form polysome. Suppose it is a messenger RNA. Okay, this is 5' end, this is 3' end, and ribosome is attached on the messenger RNA. When we need rapid synthesis of protein, then more than one ribosome attached to a single messenger RNA. This structure, in which more than one ribosomes attached to a single messenger RNA for the rapid synthesis of the protein, this structure is known as polysome. Okay. After discussing about the ribosome, we are going to discuss about the process of the translation. Process of translation consists of 4 steps. These are activation of the amino acid, step number 2, formation of the initiation complex, step number 3, elongation, and the step number 4, termination. We will discuss about these 4 steps one by one. Firstly, we are going to discuss about the activation of amino acid. In this step, amino acid combines with the ATP, join with the ATP, and ATP release its 2-phosphate group. Then remaining structure will called active amino acid, like this. AA represents the amino acid. Okay, combined with ATP, adenosine triphosphate, and ATP release its 2-phosphate group. Okay. What will remain behind? It will be amino acid and AMP. AMP mean amino adenine monophosphate. Okay, so this is the active amino acid. Now this amino acid is active. Okay, okay. Now the second step is formation of initiation complex. Okay, uh, when in the previous step, activation of amino acid. Okay, after the formation of active amino acid combined with the transfer RNA at the 5 prime end. If I represent the amino acid like a circle, active amino acid, okay, and if I represent the transfer RNA like this, uh, okay, like this is the 3 prime end and this is the 5 prime end of the transfer RNA. Okay, and it is the active amino acid. Always it will be first amino acid that will attach to the transfer RNA will be methionine. Okay, M represents the methionine. Methionine are first amino acid attached to the transfer RNA at the 5 prime end, and it will look like this. Okay, now it is the mixture of amino acid and transfer RNA. That's why it is called immunoacyl transfer RNA. It is called immunocyte. The second step is formation of initiation complex. We know that in the formation of anything, we need its constituents for its formation. So we also need the constituent of the initiation complex for its formation. What are the constituents of initiation complex? These are: number one is messenger RNA, 5 prime end, 3 prime end. Messenger RNA has sequence of nucleotides, okay, in the form of codon. The first codon, the sequence of the first codon of messenger RNA will always be adenine, uracil, and guanine. This is the first codon, and it is the stop codon. Stop codon is present at the end of the messenger RNA. The sequence of the stop codon will always be uracil, guanine, and guanine. Okay, and second is small part of ribosome. This is the small part of ribosome, like this or like this. Okay, the third one is amino-acyl transfer RNA, which is synthesized in the activation of amino acid. Okay, this is the 3 prime end, and this is methionine, because first amino acid that is attached to the transfer RNA is always methionine, and the fourth constituent is the large part of the ribosomes. When these four constituents combine with each other, then initiation complex is formed. Okay, messenger RNA attached to the small part of ribosomes at the 5 prime end. Okay, like this. This is the small part of ribosomes, and it is the messenger RNA that is attached to the 5 prime end, and it is 3 prime end. Okay, here will be the sequence of first codon, that is adenine, uracil, and guanine. Okay, after the combination of these two constituents, third constituent will combine with them, that is amino-acyl transfer RNA. Amino-acyl transfer RNA also contain sequence of nucleotides or bases here. Okay, it will attach to the first codon, and the sequence of nitrogenous bases which is present on the amino-acyl transfer RNA will complementary to this sequence. Okay, like this. This is methionine. Okay, this is the sequence of nitrogenous bases, which is complementary to the sequence of nitrogenous bases of messenger RNA. If here is adenine, here will be uracil. Okay, if there is uracil, here will be adenine. If there is adenine, here will if there is guanine, here will be cytosine. Okay, these three structures combine. Now last structure will combine with them and initiation complex will form. Okay, this is a large part of the ribosomes. Now it will attach to this structure, and this structure is known as now initiation complex. Okay, this was the second step in which initiation of complex has been formed. Okay, now we are going to the third step, that is elongation. Elongation, it is the third step. Okay, initiation complex contain three sites. This is small part of ribosome. This is large part of ribosome. This is messenger RNA that is attached to the ribosome. Okay, and these are that three sites. This is known as A site. This is known as P site, and this is known as E site. Okay, why it is called A site, P site, and E site? Because the new incoming amino cell transfer RNA firstly enter into the A site. Okay, that's why it is known as A site. After completing the A site, it will transfer to the P site. Okay, and P site transfer it to the E site, and E site transfer that amino cell transfer RNA outside the ribosomes or the initiation complex. Okay, first amino cell transfer RNA will present already because it was attached here in the initiation complex. It is the part of initiation complex. That's why it is already present here. First amino acid will always be methionine. Okay. Now suppose many other amino cell transfer RNA are present outside of the ribosomes, like this. Suppose it is alanine amino cell transfer RNA. Suppose it is an other type of amino cell transfer RNA. Okay, it is an other type of amino cell transfer RNA. Okay, now amino cell transfer RNA or any other type of amino cell transfer RNA enter into the A site like this. Suppose it is alanine amino cell transfer RNA. Okay, now an peptidyl enzyme. Okay, peptidyl enzyme put this methionine from this amino cell transfer RNA onto this alanine like this. It will be removed here because peptidyl enzyme remove it from this amino cell transfer RNA and put this methionine onto the alanine like this. Okay, now it is free. Now it is free. It is not an amino cell transfer RNA. It is just transfer RNA, and transfer RNA now enters into the E site. Okay, like this. It is free, and here it will be removed because it comes into the E site. When this site is blank, then this amino cell transfer RNA enters into the P site like this. Alanine, methionine. Okay, here it will be removed because it enters into the P site. Now new amino cell transfer RNA enter into the A site like this. Suppose this amino cell transfer RNA enters. Okay. Now peptidyl enzyme again remove these two amino acids from this transfer RNA and put these two amino acids on new incoming amino acid transfer RNA like this. It is alanine, it is methionine. These two amino acids will be removed here. Now it is also free, and it is also free. It will excite, okay? It will be removed out to the initiation complex, and this transfer RNA enters into the A site, okay? This will be removed and it will be come into the E site, okay? Like this, and this site will again be empty, okay? When P site will empty or blank, the new incoming amino acid transfer RNA enters into this site like this. This is any other type of amino acid, okay? This is alanine, this is methionine. When it enters into P site, it will be removed from the A site, okay? Like this. When it is blanked, then another type of amino acid transfer RNA enter into the initiation complex, okay? Like this, okay? Now again peptide enzyme remove these amino acid from this transfer RNA and put these amino acid onto the new incoming amino acid transfer RNA, okay? Like this. This is another type of amino acid. This is alanine and this is methionine, okay? And these three amino acids are removed from this transfer RNA. Now it is free, it is also free. This transfer RNA will be removed from the initiation complex, and this transfer RNA will enter into the E site, and P site will remain again empty, and this amino acid transfer RNA, which contain 4 amino acids, enter into the P site. In this way, this process will be continuous again and again, okay? And long chain of polypeptide chain will synthesized, and we know that polypeptide chain is known as protein, okay? When the end part of the messenger RNA enters into the initiation complex that contain stop codon with the sequence of uracil, guanine, when it enters into the initiation complex, then no any other amino acid transfer RNA enter into the initiation complex. In the place of amino acid transfer RNA and releasing factor enter into the initiation complex like this. Suppose it enter into the P site, okay, like this. This is amino acid, this is another amino acid, this is alanine, and this is methionine, okay? Here it will be removed because it enter into the P site. Now it is empty space, and if amino acid messenger RNA, the stop codon of the messenger RNA has entered into the initiation complex, what is the sequence of stop codon? Uracil, guanine, and guanine. When this sequence enter into the initiation complex, means stop codon enter into the initiation complex, then no any type of amino acid transfer RNA enter into the initiation complex. Instead, in the transfer of amino acid transfer RNA will occur the transfer of releasing factor, and releasing factor attached to it like this, okay? When releasing factor attached to the stop codon, it will disassociate in initiation complex into its constituents like these 4 constituents: messenger RNA, small part of ribosomes, amino acid transfer RNA, and large part of ribosomes. In this way, formation of protein will be stopped, and this whole process will be stopped. Translation has occurred now because our desired protein has been synthesized. So this is the lecture which I was delivered you. I hope you all understand it, and I request you to subscribe my channel and like my videos and share it with your friends. Assalamu alaikum.